everybody! So as you can see, we upgraded our setting a little bit
because the school kick us out at certain time.
Yeah, it wasn't cool.
Anyways, this is our last video on genetics.
Woo! Let's make this the best darn video about gene expression ever!
Yes, it's going to be hard, but challenge accepted.
Woo! Bring it on!
Because we've had the greatest videos on this planet.
Yes, we have.
Better than Crash Course.
Yes, better.
Actually, no, but shh, don't tell me.
Subscribe! In this episode, we'll be talking about how genes are expressed and regulated. Now, transcription has to do a lot with this process.
So feel free to review the previous episode if you get lost.
In bacterial cells, genes that are related to the same function are on an operon. And in order for genes to be expressed, a substance is needed. Now, this form of gene expression can either be controlled by positive or negative control. Repressor. So in order for genes to be expressed in bacterial cells, a substance is needed. Now, this substance determines when genes are expressed and when genes are not expressed. And the amount of the substance also determines when genes are expressed and when genes are not expressed. Now, positive control and negative control are both ways that gene expression is regulated, and we'll get to that later in this episode. Now, repressors and inducers are both involved, and we'll also get to that later in this episode. Repressors inhibit gene expression, and inducers are what either cause repressors to stop inhibiting or inhibit gene expression.
That's like in India when a guy hits on you. The repressor is like the ugly guy who is like very creepy and you don't want to be with, so you try to run away, okay? But in India, the inducer is a very, very hot guy, so hot that he will denature your enzyme, okay? And you feel so attracted to him, so you like, okay, I will let you control my operon. Johnny Lever is like the repressor, and John Abraham is like the inducer, because John Abraham hot and then John Lever like not.
I have no idea who these people are, and I doubt that anyone else will.
Okay, think about it this way then, okay? Ice JJ Fish, repressor, and Bruno Mars is inducer.
Ooh, Bruno Mars! I like the sound of that.
Yes, true.
Anyways, with negative control, the repressor normally blocks the path of the transcription factor, and an inducer is used to make the repressor fall off of the transcription factor's path. Now, an example of this is the lac operon, in which lactose binds to the repressor and makes the transcription factor be able to express genes and make proteins.
So that's like the ugly guy hitting on you again, okay? He hold on you so tight, okay, that you can't even get away, but the police officer finally gets on to him and he pulls him away and you good, okay?
Yep.
And that ugly guy's arrested.
So positive control is the exact opposite of negative control, of course. The repressor normally is not binded onto the path of the transcription factor, but the presence of an inducer will make it bind to the transcription factor's path and prevent gene expression and the creation of proteins. An example of this is the trip operon, where tryptophan, which is found in turkey, binds to the repressor and makes gene expression not happen.
This is when you're talking to the cute guy and then you having a great time, you hold your hand, but then your cuter, funnier, smarter cousin walk up and then he get attracted to her, so he go away from you and she takes him away, okay? Stupid, this happened to me. Oh, shoot, my mommy doesn't need to know.
So that was easy to understand, right? Well, it's about to get a lot harder. Because now we have to talk about eukaryotic gene expression.
No, we don't. It's the same thing, right?
No, not even close.
Why don't we just start into bacteria cells?
Well, you really don't like eukaryotic gene expression, do you?
It's too complicated.
Yeah, no, I don't blame you.
So there are many things that regulate gene expression in eukaryotic organisms. The first one is the presence or lack of a protein that is bound to the strand of DNA, and that affects the transcription factor's ability to bind to the promoter region. The second thing is when introns are removed from a strand, it allows for just the exons to be there, and it allows for a shorter strand, which means that genes can be expressed faster and more proteins can be produced. The third one is the ability of this mRNA strand to exit the nuclear membrane via channels. The fourth one is the presence of enzymes around the nucleus that can break apart our mRNA. And the last one is if the proteins outside of the membrane have been chemically modified.
You got an analogy for that? No, I don't understand this one. Well, is there anything else to talk about? Oh yeah, biotechnology. Oh, son of a bacterial chromosome. Exactly. Speaking of that.
Plasmids are a certain unique structure in a bacteria cell that, in addition to the bacterial chromosome, the plasmid actually allows evolutionary traits to be passed down to other bacteria cells. Okay, so the plasmid can be passed down from different bacteria cells via a conjugation pili, and that is so cool, right? And another thing called natural selection allows the bacteria cells without the resistance in the plasmid to die, while the bacteria cells with the resistance live. So more bacteria cells are resistant to the antibiotic. Yes.
It's like when you're the first person to take a test in a class and you realize it's really hard, so you warn all of your other friends about it beforehand, and they end up doing really well. Also, your class then ends up failing because you didn't have that advantage. Except do this after college because cheating is bad.
Yeah, actually don't do it at all. Wait.
Yeah, don't do it. Forget we said it. Okay.
Oh wait, when did we switch jobs?
Oh, whoops. But you seem to have the hang of this, so go on.
Wait, really?
Yep.
You know what? You should take money out of this shame jar. Use it on yourself. You good boyfriend.
Nah, we're broke. Remember, we need it for the studio.
Okay, so one method used to manipulate the traits of a bacteria cell is by using a vector. A vector is basically taking a restriction enzyme and cutting the DNA of a eukaryotic cell or any other cell and inserting it into the plasma of a bacteria cell. The plasmid is then put into the bacteria cell, and now the bacteria cell has these supernatural cool traits that allow them to be resistant to things and do things differently. Oh, they cool. Yes.
Apheresis and short tandem repeats. It can also be replicated really quickly using PCR or polymerase chain reaction to denature them and then cool the DNA sequences. So
Wait, are we done?
Yeah, I guess.
Wait. We're done. I'm never going to get over this.
Hey, calm down. Hey, we might be back for other videos. I mean, after all, this was just a school project.
Wait, really?
Yep.
Oh, okay. But this was so much fun and now it's already over.
Well, I mean, if we get famous enough from this, I don't see why we can't continue.
Okay, let's tell our friend the thing we were planning.
Oh, yeah, okay. Make like mitochondria and be the powerhouse of yourselves.
Hey guys, thanks for watching our videos. It's been so nice to get to know you all and you guys watching our videos, and thanks to our AP Biology teacher for letting us do a crazy final project.
Yeah, you're the best, and thanks for being such an amazing audience, you guys. You know, this is our last video because this was a school project, but if you want to see more of us, then just like and subscribe and we'll make more.
And also, we have websites and a quiz and everything to help you with genetics, so if you're struggling with genetics, we have that in the link in the description below, so make sure you check that out.
Yeah, we have a test up for genetics too, and if we do more, then we'll make more tests. We have a blog and a forum, well, a blog, and yeah. We're kind of tired, but we have a forum, and if you guys ever want to talk, we'll be on there all the time. And if you guys just want to talk with each other too, that's cool.
And yeah, so it's been great, and we'll make more videos if you would like them.
Yeah.
This is just per request, but this was mainly for a school project.
Yeah.
So yeah.
So I guess what we have to say. Oh gosh, this hurts.
I know, right?
This has been Binalogy, where I taught you biology.
And I taught you the analogy.
So you got it. Bye, y'all.